All right, it is 11 a.m. here in Texas, so we're going to start our presentation. Hello, everyone. My name is Andrea Botazatu. I am an assistant professor and Enology extension specialist with Texas A&M, Texas AgriLife Extension Services, and this is our Enology webinar series. Before I introduce the topic today and the presenter today, I just wanted to go through a little bit of housekeeping. This presentation will take about 40 to 45 minutes. There will be a Q&A session at the end. So if you don't mind holding your questions until the end. Alternately, you can type your questions during the presentation in either in the chat box or the Q&A box there, and we will try to answer them all at the end of the presentation. Also, I am located in Texas, so you might have heard, maybe yes, maybe not, but there's a big winter storm in Texas happening right now. Temperatures are very low, the power grid is overloaded, so there is a chance that the power might go down during this presentation. If that happens, please try to log back in. If we can't log back in about 5 to 10 minutes, then we'll try to reschedule the webinar. But this is totally out of our control. So doing our best here to try and weather the storm. Also, there will be a very short survey at the end of the presentation. I would really appreciate it if you took 1 minute of your time to fill out that survey. That survey allows me to continue my webinar series, and it allows my bosses to rate me and rate the quality of my programs. So if you like this, please help me out by filling that little survey at the end. All right. So now it is time to introduce our topic and our presenter. And I'm very, very excited about today's topic. We're going to talk about the Aromalock method for recovering and preserving volatiles during wine fermentation. And as a former commercial winemaker, I was thrilled to hear about that and thrilled to hear about the potential of this. So I was really looking forward to sharing this new development. Our presenter today is Dr. Dick Jones, who is the inventor of the method. Dr. Jones has a PhD in pulmonary physiology and worked as a professor in the Department of Medicine at the University of Alberta, Canada for 38 years. He also worked as a bush pilot in the Canadian Arctic, invented nicotine nasal spray, and grew wild rice in remote northern Alberta. He retired to British Columbia, where he grows grapes and is an amateur winemaker. He invented the Aromalock method to preserve fermentation aromas, and his presentation will be wine aromas and how his method can help preserve them. So Dr. Jones, please unmute yourself at this point and take it away.
Hey, so my name is Dick Jones. I invented a process to ferment wine called Aromalock that preserves many of the volatile aroma compounds that are produced during fermentation. I have never taken a course in enology or viticulture, but I am a scientist. I had almost 40 years of doing research on the lungs, and I was a professor at the University of Alberta. In 2005, I retired, and my wife and I came out to British Columbia and bought a house here on 4 acres of land. And it didn't take too long before I got caught up in the wine culture that's quite prevalent around here. Where we live is about 10 miles from a little town called Penticton, and that's where we go to get groceries and so on. But in that 10 miles, I'll pass 40 wineries. And so these are all boutique wineries, small. There's not a lot of land space to use for growing grapes, so the output of each winery is relatively small in this area. But still, there's great pride in producing quality wines in the Okanagan Valley, and I got to know many of the winemakers. wondering what I was going to be doing for most of my retirement. I thought, being previously a very active person, I would plant a little vineyard for myself. I enjoy drinking wine, and all of our visitors from Alberta enjoy drinking wine, so I wanted to have plenty on hand. And so I started trellising across the hillside behind our house, and that's a very steep hill. You couldn't get the equipment in there, but I could go down with the shovel, of course. And so I made 9 terraces across the hill, got some cuttings from a friend, rooted those and planted those, and ended up with about 140 or so vines in that little vineyard. And then I planted some more along that walkway going down to the vineyard. So as you can see, perhaps the soil isn't that good, and it took some time for the vines to establish themselves. This was in 2006, but a few years later, they were going reasonably well. And I had my first harvest, I think it was 2009. During that harvest and winemaking season in 2009, I made only perhaps 100 liters of wine that year. But as the years went on, I made more. And by 2012, I had some pretty robust vines, and it was producing some really nice grapes. In 2012, when I was fermenting the wine, I happened to walk by the fermenter in my tiny little winemaking room. I tell my wife we should call our place the land of little. Everything is little here. It's a big piece of property and a big view, but the vineyard's tiny, the garage is tiny, the winemaking room is tiny, and so on and so forth. So that fermenting wine was sitting on the counter, and I went by and I smelled the exhaust from it, and I said, "Whoa, this is going to be just an excellent Pinot Gris." And then I went out and did something. I was cleaning up the garden, I think, and I realized that, wait a minute, all those aromas that I smelled coming out of the fermenter should stay in the wine. I'd like them in the wine. And so I did some thinking about it and wondered just how much aroma is lost from fermenting wine when the CO2 blows it out. So I did a literature search, and this is some of the first material I found, and it's maybe still the best material. A fellow named Ferreira in Spain did a study back in 1996, and his data is shown by the blue bars on this graph. He studied 3 groups of aroma compounds, a fusyl alcohol, isoamyl alcohol, an acetate ester, isoamyl acetate, and an ethyl ester, ethyl hexanoate. And he found the alcohol didn't decrease very much, and so the CO2 didn't blow it out very much. Only about 10% was lost in his study. But the esters, both acetate and ethyl esters, had some pretty significant and large losses. So they were up around 50%. So he showed then that the loss of aromas is significant in wine. And I thought, I'm going to try to overcome that with something. I then found another paper, which is shown by the red bars. Ferreira did his study by bubbling CO2 into a synthetic wine solution. The red bars are from a French group that used an actual fermentation and developed a way to measure the amount of aroma produced, the amount of aroma lost, and the amount of aroma retained in wine. And the red bars show the losses of aroma. Again, the isoamyl alcohol was essentially not lost at all, but the esters, both acetate and ethyl esters, were lost to a fairly significant degree. You see that the ethyl hexanoate was up around 45% loss. and the isolamyl acetate about 25% loss. These losses get bigger if you do a warmer fermentation. This particular graph was obtained at 20 degrees for both of these authors. As you warm it up, ethyl hexanoate can be lost to the tune of about 75 or 80%. So loss of aroma is a significant thing when we're fermenting wine. Ferreira put a statement in his paper saying that up to 80% of the volatile material can be blown off, while an average of only 10% is retained. So the wines we're drinking now really are a shadow of what they could be. The aromas are lost to a large degree, and well, to be frank, I wanted to see just how important these aromas were before I gave much thought to a way to prevent it. So I did some more literature searches, and every paper virtually that I read from an aroma scientist had a statement somewhere in the introduction or in the conclusions of the paper. For instance, and I put three of those here. Molina in the early 2000s said that as far as consumers are concerned, the aroma and flavor of wine are among the main characteristics that determine its quality and value. Then Moret in France, in his group, in a paper said, wine aroma is one of the principal attributes determining consumer preference. So, okay, I'm becoming a believer that aromas are very important. Then a graduate student at Virginia Tech University said in her master's thesis, winemaking techniques that would function to improve the profile of desirable volatile compounds would be beneficial in the creation of high quality wines. So if there was some way to not only prevent aroma loss, but especially prevent the loss of desirable aroma compounds, that would be a big boon to the wine industry. So I went away and thought about ways to do it. Being a pulmonary scientist, I dealt with gases every day, and I was comfortable thinking about how gases move and whatnot. The first paper I published back in 1969 was on a membrane I developed for oxygenating the blood. So it didn't intimidate me to think of ways that we could retain these aromas. But before we get into what we did with Aromalock, I'd like to just review for a few minutes aromas, and it'll be old hat to many of you who work in this area, but I think it's worthwhile just driving home the importance of aroma and the volatility of aromas. So here we have a lady who's sniffing a glass of white wine, and you see there's an aroma compound, a red one, up in the airspace above the wine. Now she's going to sniff that gas into her nose, and it's going to carry that aroma compound with it. The aroma compound is going to lodge on the nasal epithelium and create a reaction depending on which receptor it hits. And there's only a limited receptor choice for these aroma compounds because they're kind of like a lock and key. Their shape fits into only certain receptors. But when the receptor is activated by the compound, an electrical signal is sent to the brain, and then you recall something that you smelled before that's reminiscent of that compound. In this case, it might be a banana, so it could be something like isolamyl acetate, for instance, that is the important compound for the banana smell. You'll notice that after she inhaled that aroma compound, another one left the wine and went up into the headspace, ready for the next sniff from the glass. You also notice in this illustration some blue aroma compounds that are in the wine, and none of them are in the airspace above the wine. We would say that these aroma compounds are not volatile. In other words, they don't escape from the wine and do not enter the gas phase. We'll never know those compounds exist unless they're free to move and we can inhale them. Now, the other way, of course, that you can smell aromas is to sip the wine and swallow it. And then when it gets to the back of the throat, lots of aromas are released because of different properties of the saliva. And of course, we're warming up the wine as we put it in our mouth. That makes all aroma compounds more volatile. So these can come up and hit these same receptors in the nose, but do it backwards by the throat. And they come up and stimulate the appropriate receptors and tell us what that aroma is reminiscent of. So what is volatility? And I think this is something that's very important. If we did a little experiment and had two beakers, beaker A and beaker B, both of them had some water in them, and then we took a pipette and put in a concentration of an aroma compound into each of these beakers, and then put the lid on and let the compound just equilibrate between the air and the water. And after an appropriate amount of time, we would measure the compounds in the air and in the liquid in the water. And in example A, beaker A, you see there's one compound in the air, but 30 in the water. So the gas to liquid ratio is 1 to 30. In beaker B, a different aroma compound, still has 30 for concentration in the water, but you see now there's quite a bit more in the airspace. There's 6. So the gas to liquid ratio is now 6 divided by 30. So we would say that this ratio is much higher in beaker B. Beaker A we would call a hydrophilic aroma compound because it likes staying in the water. In beaker B, we would call a hydrophobic aroma compound because it doesn't like staying in water and tries to escape into the surrounding air. So this gas-liquid ratio is called the partition coefficient, and it gives us information about the volatility. So hydrophilic compounds generally have low volatility, and hydrophobic compounds have higher volatility. Well, the reason I'm bringing all this up is because you're going to see later on when I talk about some results, a value called log P, and that's just the logarithm of the partition coefficient. So the higher the value for log P, the more hydrophobic the compounds are, and the important wine aromas tend to have higher log P values. So you're going to see log P again later on. Let's just talk for a second about what the yeasts do. Of course, we all know that the yeast produce a whole bunch of carbon dioxide by metabolizing the sugars from the surrounding liquid, and they also produce ethanol. And ethanol, as it accumulates inside the yeast cell, will eventually reach a concentration where it diffuses down its concentration gradient to the surrounding liquid. However, some of the ethanol is used for other purposes inside the yeast cell. For instance, I show here ethanol, ETOH, combined with hexanoic acid, a fatty acid, can produce a different compound called ethyl hexanoate. Now, ethyl hexanoate is called an ethyl ester, and it has an apple smell and so-called taste to it. It's one of the important aroma compounds in wine. So we take two things that don't have very much odor and combine them and produce something that has a whole lot of odor, in this case, ethyl hexanoate. Now, I'm going to use that compound for a lot of my discussion here, but believe me, there are a lot more than ethyl hexanoate that's produced by the yeast cell. There might be 800 to 1,000 different aroma compounds produced. They're usually all produced by combining something with something else. So the little yeasts are doing a big job here. Besides just producing CO2 and alcohol, they're producing a whole bunch of higher alcohols and ethyl esters and acetate esters and other things as well. Okay. Anybody who's studied fermentation at all has seen a graph like this before. It shows the CO2 production after a wine has been inoculated with yeast. So at first, over on the left side of the graph, you see nothing's happening. There's no CO2 production, and we call that the lag phase. Don't believe that there's nothing happening during this phase, though. The yeast are actually very busy, but they're not busy producing CO2. They're busy producing other yeast cells. So the density of the yeast in the liquid is rising during this lag phase. Eventually, the yeast population gets high enough where the yeast shifts its attention from making babies to producing CO2 and alcohol. And now you can see the CO2 starts to appear and will rise rapidly to a peak and then fall off slowly over a period of time. The fermentation time in this case might be a week or 2 weeks, something like that, depending on the temperature. So when are these aroma compounds produced? Knowledge from papers published by the French group show us very nicely when aroma compounds are produced. Ethyl hexanoate, for instance, starts being produced just after the appearance of carbon dioxide. And the reason it's delayed is because we have to have some ethanol in order to combine it with the hexanoic acid. We need those two things before we can get ethyl hexanoate. So we need some ethanol around. So ethanol builds up inside the yeast cell, and then when it reaches a certain concentration, it can then diffuse to the outside of the cell into the liquid of the wine, or it can be used from inside the cell to produce aroma compounds. So the lag phase is simply the result of requiring one of the substrates for ethyl hexanoate to be available, and that is ethanol. But anyway, that's when the ethyl hexanoate is produced. If we looked at it another way, this is the rate of ethyl hexanoate production. The first line, the solid line, was the total production of ethyl hexanoate. So this is the rate, how much ethyl hexanoate is produced per hour, for instance. And you see that it kind of follows a pattern similar to the CO2. It rises rapidly to a peak and then falls off during the fermentation. So most of the ethyl hexanoate is produced pretty early in fermentation, and then it slows down later during the fermentation. So what is the problem that I eventually tried to address? Well, you've heard about it already. This slide shows a tank of must being fermented, and the must contains some ethyl hexanoate in the liquid and some in the headspace or the gas phase above the wine as well. Now, this would be an example, let's say, at the point marked X on the fermentation time. So it's pretty early in fermentation. There's a lot of CO2 production, and there's also been quite a bit of ethyl hexanoate produced already. So ethyl hexanoate is building up in the liquid, and because of that gas-liquid partition coefficient, it would like to escape to establish whatever it is for that compound. And so ethyl hexanoate will leave the wine and go to the headspace. The problem is there's so much CO2 coming out. It's a constant flow of CO2 that if you're a little molecule of ethyl hexanoate in the headspace, you're kind of a sitting duck. The CO2 is just going to move you right on out into the winery. And that's when we smell these aroma compounds in fermenting wine. If you've ever, some of you may not have, some of you certainly have been in wineries when wine is fermenting and you smell the aroma compounds. Well, this is what you're smelling. You're smelling these aromas that have escaped from the wine, gone to the headspace, and were blown out to the winery by the CO2. So I wanted to see if I couldn't find out some way to prevent that. But I had some guidelines actually when I was thinking about the methods I might use, and they all had to be non-invasive. I was very familiar with many of the winemakers around this neck of the woods, and I soon realized that they don't like to fiddle too much with the wine. And I thought, well, if I'm going to take the time to invent something and eventually try to bring it to the market, I don't want something that winemakers won't use. So I'm going to try to keep this totally non-invasive. I'm not going to touch the wine in any way. So I came up with an idea, and I'll be explaining that in the next slides. So this is the problem. Ethyl hexanoate is produced in the juice, but it doesn't stay there. It moves to the headspace, and then it's blown out into the atmosphere. In other words, you've lost aroma from the liquid wine. Now, fortunately, something happens during fermentation that's beneficial to keeping the aromas in the wine. And that is, as we go further and further along the fermentation time, of course, more ethanol is produced. So the wine's gaining ethanol. And the other thing that's happening is the sugar concentration is dropping because the yeast have gobbled it up. So these two things, alcohol and loss of sugar, tend to make aroma compounds more soluble in the liquid, which is a good thing. So toward the end of fermentation, even ethyl hexanoate won't go too rapidly to the headspace, not like it did early in fermentation. So the loss of aroma slows as we go toward the end of fermentation because of these changes in what's called the wine matrix, the juice matrix, the composition of the juice. So in this graph, we see the ethyl hexanoate, the blue line that is produced, is nowhere near the ethyl hexanoate that is retained in the wine, the red line. What's happened to it? Well, most of it's due to this phenomenon of Co2 aroma stripping. So we lose quite a bit of the aroma during fermentation from this simple mechanism. Now, all volatile compounds are susceptible to this, some of them more than others. I give you the example of ethyl hexanoate. Well, that's just a moderately volatile aroma compound. There are aroma compounds much more volatile, and they are lost to a much greater degree. There are some that are not lost very much at all, like the fusel alcohols. They're pretty well retained in the wine. So what happens when we're losing all these desirable aromatics is that the aroma profile of your wine is shifting. It's shifting toward more weight from the fusel alcohols and less from the fruity esters. And that's the wine we're accustomed to drinking. So let's just take a look at this. Sure, CO2 aroma stripping causes a large amount of aroma loss, but each post-fermentation processing step adds to this loss. And I'll just show that to you. It's something a lot of people don't think about. In fact, when I started out on this, I just thought, things stopped at the end of fermentation. Well, they don't, because of course we do a lot of fiddling with the wine afterwards. We rack the wine. We maybe use bentonite clay to fine the wine to remove proteins. We maybe filter the wine, sometimes with very fine filters to get rid of bacteria and so on. Eventually, we end up bottling the wine. We then let the wine sit somewhere in a storeroom before it comes out on the shelf for someone to buy and drink. So there are a lot of steps in between, and there's some pretty good data now recently on how these individual steps can affect the aroma compounds. This is not meant to be a precise representation of what happens at each step. I just show a steadily decreasing aroma concentration with each step. These decreases from step to step depend on how sloppy the winemaker is. If he's going to splash the wine around during the racking, he's going to lose more aroma than if he or she is very, very careful. If you use too much bentonite clay, you're going to lose more aroma than if you use just the minimum amount. And if you filter once or maybe even twice and use fine filters, you're going to lose more than if you don't filter at all. And the act of bottling may actually cause some aroma loss as well. Well, then we store the wine for a few months at least, and you see the aroma concentration continues to deteriorate. And this is due to a process called hydrolysis. So if it was ethyl hexanoate in the wine, we would find that that would decrease with time in the bottle. And the reason it decreases is there's a natural reaction of esters where they break down into their component parts. So ethyl hexanoate would be broken down to some degree over time to ethanol and hexanoic acid. Well, you see the dot at the end of this line is where that consumer is going to taste that wine. It's pretty far removed from the total that was produced by the yeast. Now, is it important to try to correct this? Well, one of the French researchers, Moret, said in the last paragraph of one of his papers, ethyl hexanoate losses approached 40% at 20 degrees centigrade. This loss can have a direct effect on wine flavor. Okay, so you would certainly guess that if there's a 40% loss in something, it should affect the flavor. But then he went on to say, if there was a way to regain that 40% concentration of ethyl hexanoate at the end of fermentation, then that's very important because it will give us a higher concentration in the bottle after storage. So yes, it's very important to try to prevent the aroma loss during fermentation because the higher the aromas going into the bottle, the more you're going to have after storage. So what was my idea? Well, I show it here. I have a tank of wine that's being fermented. There's liquid wine down below in the gray area, and then up above that is the headspace. Now there's CO2 being produced, so that's going to push gas out from the headspace because the pressure develops in there. If we tap that headspace and duct that gas to an Aromalock machine that contains a special membrane, then what we're going to do is feed that membrane gas from the headspace. So that's the feed gas going to the membrane. As the feed gas flows past the membrane, any aroma compounds or CO2 as a fact will diffuse across the membrane. Its diffusion across the membrane is assisted by creating a vacuum on the other side of the membrane. So we have a little vacuum pump in there that's sucking. That's going to help aromas move across the membrane. Anything that doesn't go across the membrane is going to exhaust. In membrane terminology, that's called the retentate. It's the stuff that's retained after gas has moved through the membrane. Okay, so what I did recently, and this was done, I think it was early December of a few months ago, to test different parts of our Aromalock circuit in an experiment I did here at home. It was right behind this wall, as a matter of fact. I set up an Aromalock system and took samples of the gas in the feed, in the retentate, and in the permeate parts of the circuit, and took them to our local college, which has a gas chromatograph mass spectrometer set up and can measure aroma compounds. Especially in gases, it's fairly easy for them to do. Collecting them is another thing. It's not so easy to do and keep them from leaking as you drive into town with them. So this is an example of the data I got. So in the feed, there's in the retentate, there was also a little bit of ethyl hexanoate. In other words, we weren't 100% effective in removing all the ethyl hexanoate in this particular experiment I did. However, it was an experiment done at a very high flow of gas through the membrane. So it's kind of overwhelmed the membrane. Still, there wasn't much left. I chose this particular example because there was a bar for the retentate, the red bar. Lots of other compounds show no bar. So this was ethyl hexanoate that had been lost out to the Co2 winery. Well, where'd the other stuff go? Well, it went across the membrane. Now, remember that a lot of the aroma is going across the membrane, but only a little bit of the CO2 is going across the membrane. Perhaps 10, 20% at most of the CO2 is going across the membrane. So there's not too much CO2 in the permeate stream that's being pumped back to the headspace. So this makes for a high concentration of aroma in the permeate gas. You can see it here. It's approximately 4 or 5 times the level of the feed gas going to the membrane. That's called the enrichment factor. So that would be an enrichment factor of 5, let's say. So the permeate is 5 times more concentrated than the feed. Now that concentrated permeate gas goes back and we duct it in close to the surface of the wine, and it theoretically spreads out along the surface and makes for a high concentration of aroma there. Now remember, inside the wine is our little yeast that's busy producing ethyl hexanoate. And once a ethyl hexanoate is out into the juice, it's going to want to move toward the headspace because it's volatile. It is hydrophobic. So as it's produced, it starts heading to the headspace and says, whoops, the concentration is too high in the headspace. So I'm just going to turn around and come back and stay in the liquid until I build up enough concentration in the liquid where I can move then to the headspace. So this is the way the Aromalock system works. It does so by simply modifying the headspace aroma concentration to prevent aromas from leaving the wine, not necessarily to force aromas back into the wine from the headspace, although that may happen. It may happen as well, but I'm kind of skeptical about that because, as you know, these desirable aromas are hydrophobic. They don't like to go into the liquid. So at the end of a fermentation using Aromalock, we may get a ethyl hexanoate concentration in the wine that is considerably higher than the one you saw before, maybe a 10% loss. And of course, after we do all the stuff at the end and lose some more aroma, we end up with a wine that has more aroma than if we didn't do anything, if we just use standard fermentation practices like they have been conducted over the last 8,000 years. Okay, so I'm sure you want to know what kind of results we get. Well, I'm going to show 3 graphs. And this is data that just came in the last few months. I'm always quite a fan of presenting your latest stuff you have. So this is data from wine that was fermented using the Aromalock system, and also with a exact control wine fermented alongside it. Now the control wine is always done in the standard way. There's an outlet to the atmosphere. The Aromalock wine has the headspace ducted to the Aromalock machine, where it goes, the aroma compounds go across a membrane and go back to the headspace. Okay, so here's what we found. Each dot on this graph is a different aroma compound. And these were measured at Brock University in St. Catharines, Ontario in Canada. It's our viticultural university here in Canada. And they do quite a good job at these measurements. So this was an example of a Pinot Gris fermented last October here in the Okanagan Valley of British Columbia. And that wine was bottled, but not filtered. Okay, so this is Pinot Gris that's only a few months old. We sent samples of that along with its control down to Ontario for this analysis. And like I say, we just got this data back. I believe this was data that I received toward the end of December. It shows this. It's a graph of the Aromalock concentration of the aroma presented as a percent change from control. So any number below 0 means the Aromalock concentration was lower than the control concentration. Any point above 0 means the Aromalock value had a higher concentration for that aroma than the control wine. And you can see there's a linear relationship between these points plotted against log P. Remember, log P is the log of the partition coefficient. So how hydrophobic are these compounds? We had a nice spread of hydrophobic values for these compounds. So we got points spread out along the x-axis quite nicely. So the low values for log P mean the compound is very hydrophilic. It's not very volatile. Higher values for log P represents compounds that are hydrophobic and are quite volatile. Well, you see what Aromalock tends to do. It tends to preserve the hydrophobic compounds, those on the right side of the graph, compared to those on the left side. They may even be not changed or lost to some degree. So what Aromalock does is changes the aroma profile of a wine. It doesn't increase the fusel alcohol levels, but it does increase the aromatic ester levels in the wine. This is a good thing in my opinion. You'll see some tasting data in just a couple minutes. Now here's another graph set out the same way with the log P value on the x-axis and Aromalock versus controls on the y-axis. And this looks very similar to the other graph. So this is a red wine, and this was fermented in California 2 years ago, in 2019. And we just got those samples, I think it was in the late fall. So we were able to send them down to Brock University for aroma analysis. So this is fresh data, but in almost year and a half old wine. And it shows a similar pattern though. The ethyl esters, the acetate esters that are hydrophobic are preserved and increased in Aromalock treatment, but those with very little volatility, like the alcohols, are not conserved with Aromalock treatment. Now, this is an interesting study because it was a red wine, and the whole premise of doing the Aromalock procedure is to maintain an airtight headspace so that gas coming out of the headspace is going only to the Aromalock machine and is being pumped back into the headspace. So we need a gas-tight headspace, ideally. If you have a big leak around the gasket of a floating lid tank, for instance, you're going to make Aromalock much less effective. So we had to rig up a special system for this wine, make it a stainless steel head to replace the wooden head. And that stainless steel head had a built-in punch down tool. So we could do punch downs without opening the wooden barrel. So it had several challenges. First, it was a wooden barrel, which are harder than heck to make gas tight. And it had to have a punch down tool that didn't allow air to escape when you did punch downs. So we solved this problem, and this is the result. We got the people at this winery in California did send some tasting data, and that showed universal. I think it was only 4 tasters. They couldn't get people together either for tasting, but showed universal opinion that the Romalock one tasted better. Now, I'll show you one final graph, and they all look pretty much the same. This is a Pinot Blanc fermented over a year ago, and that was done here in the Okanagan Valley. And this Pinot Blanc was notable because tasters thought it was just really outstanding. And we let this wine sit in the bottle for a little over a year before sending samples of it down for GC mass spec analysis. So this came back in January, last month. This data arrived back from Brock University, and it shows almost identical pattern to the other ones. Even though that white wine is over a year in the bottle, we still see the same pattern. The aromas were more concentrated if they're hydrophobic aromas, and that's good. Now, it's hard to compare this to just after fermentation. We did do studies at that time, but it was done in a different laboratory at the University of British Columbia. Has a very good Nobel Prize winning GC mass spec lab. But we analyzed different aroma compounds than the ones that we studied at Brock, but the pattern was exactly the same. 40 to 60% changes in the most volatile compounds were observed when this wine was sampled right after fermentation. And they're still up there, holding up to 40% changes in some of those aroma compounds. So I think Moret was right, the Frenchman, who said, if you go into the bottle with more aroma, you're going to have more aroma at the end of the whole process when the person tastes the wine. So that's a good thing. So what about tasting? It's one thing to look at a bunch of dots on a graph, and it's quite another to see if it's any good or not. Theoretically, if you have more hydrophobic aromas, you should have more fruity taste in the wine. This is a Pinot Gris that was tasted by a fellow who lives just down the road from me here, who is a master of wine, a highly sought after guy around here. He's a pretty good taster, and he tasted this wine, as did 16 other winemakers, and it showed a similar response for the whole group. But I picked his out because I thought it was interesting. He showed that all of the attributes that we tested for, fruity aroma, varietal aroma, and so on and so forth, were increased about 20% or so in the Aromalock wine compared to the control wine. So the control line is the red line on the graph, and that's just artificially set at 0. So anything that's better than 0 is outside that red line. Anything that's worse is inside the red line. Well, you see that all the points here were outside. But the thing that struck me most about his assessment was the big improvement in aftertaste. And I noticed that myself. I'm getting up in age, and my taste buds may not be too good, or my nasal epithelium may not be as sensitive as it once was. But this is what I see when I taste a wine made with Aromalock. I get this lingering aftertaste of aroma continuing to come up, retrograde into the nose, into the nasal epithelium, and you have this sensation of fruity aroma for a long time after taking a sip of an Aromalock wine. Here's another one that was just before the COVID lockdown. We had 22 winemakers sample one of our rosés that was fermented with Aromalock. And you see, that's kind of a boring comparison. It's just everything was increased about the same amount. Quite a bit of a change from the control. So all the tested attributes were increased. Now, that's fine, but you're going to ask me, if you're in Texas, does it work in Texas? Well, fortunately, we had a winemaker in Texas participate in a trial we did just last fall. We sent him an Aromalock machine that was to be used, and part of the deal was that he would send samples back for us to analyze. And this winemaker, Jason, I think, is probably on this webinar. Maybe he can comment later, but he tasted it. I hope it was blind taste, but this is what he found. And it was just one tasting, but it suggests a pattern consistent with the other tastings we've become accustomed to for Romalock. So it seems to work in Texas too. Now, that's all I have to say, but I would be remiss if you thought I did all this by myself. I have a really good group of people working with me in Aromalock. I have Paul Gardner, who's a professional winemaker and engineer, and he's our CEO of Aromalock. Walter Meyer, the Swiss fellow who had a career in the banking industry, and also a very avid amateur winemaker, and worked at some of the wineries around the Okanagan since he moved here. I think it was in 2000. And then Pete Desai is our director of business development. And Pete is an entrepreneur and worked at Dow Chemicals for years and years, and successfully launched several startups over the years. And then our communications director, Randy Morris. So these are the guys who helped me. And so I'm certainly not doing all this by myself. So I want to thank you for the time to watch this presentation and to listen. And I guess there's going to be some time for questions.
Thank you, Dr. Jones. Yes, we're opening it up for questions now, and we already have one from Dietmar. He says, conceptually, I do not understand why the larger ester molecule traverses the membrane in preference to the smaller CO2 molecule.
That's a very good question. It's the type of membrane that we developed and which we use is a membrane that has no holes in it. Okay, it's called a dense membrane. It's a type of rubber, really, food grade rubber. And so things don't just kind of move through the membrane through holes. What happens is it's a two-step process. For a compound to go across our membrane, it has to first dissolve into the membrane. It has to dissolve into that rubber. And if it's going to dissolve in there, it has to be a very hydrophobic compound because our membrane is hydrophobic. So a compound will not dissolve into the membrane unless it's hydrophobic. So it can't move across the membrane unless it's actually in the membrane. And so once it dissolves in the membrane, it can wiggle its way across, whether it's a big or small molecule. Some of the smaller molecules, like the alcohols, it's a very good question. The smaller molecules are hydrophilic. They do not dissolve in the membrane material, so they cannot diffuse across. If they could dissolve in it, yes, they would zip across faster than the bigger molecules. So it's a function of whether these compounds can dissolve into the membrane material itself. So this is called a solution diffusion mechanism for gas transfer across our membrane. Solution, meaning it has to dissolve, and then it has to diffuse.
I have another question. Thank you for your response to the first one from Briggs. I must have missed this, but how do you handle undesirable volatile aromas?
Yeah, that's a very common question. Fortunately, a lot of those undesirable thiols and so on are pretty hydrophilic. They dissolve into liquids, so they don't go across the membrane to any degree. And now I have to be honest with you, we haven't really tested those. I've never spiked a sample with these compounds and looked at what happens. And all I can say is we've done perhaps 50 trials so far, 50 fermentations, and never have found any evidence, for instance, of hydrogen sulfide or thiol contamination in the finished wine. We have seen it in the control wines, especially in one cider that we used Aromalock on. Couldn't drink the control wine. It was terrible. It had some kind of a compound in that was a sulfur compound. The Aromalock cider was absolutely clean. So I just don't think these compounds go across the membrane simply because of their chemical properties. They tend to be exhausted out the end.
Thank you. If you have any more questions, please type them either in the Q&A or the chat box. I have a question for you, Dr. Jones. This machine, is it individual? So is it one piece of equipment per tank? How does it have a volume requirement? So the practicalities of this, what does it look like? How does it work in a winery? What does it look like?
Okay.
And, yeah.
Can you people still see this screen?
Yep. Yes, we can.
Okay, so this kind of gives a pictorial history. This is back in 2012. My wife snapped a picture of me. I had to sleep in the wine room because I had these flat sheet membranes here that were recirculating headspace gas, and it could easily have created a pressure problem and I'd blown up my carboys. So this is some of the first wine ever done with AromaLoc. The next one over here shows a scaled up system they used in a commercial winery, but obviously a prototype machine with membranes and pumps and so on, pressure gauges and whatnot, hooked up to the headspace of a tank. So that headspace gas is going down here to these membranes and then through the membranes and then back up into the tank headspace. So that was sort of the first iteration of our Romalock machine. And that developed into a machine like you see here in a winery. There's two machines that we built that have these membranes behind them, and that machine was wheeled in and with long hoses hooked up to the outlet of the tank. As you can see, for instance, it's easier in this one probably to see the blue hose going up to the headspace outlet. So headspace gas went to the membranes, it was treated, and then there's another tube in there for it to go back up to return to the headspace. So this machine, although it worked, ate up quite a bit of floor space. And they're big long hoses. I mean, we were learning as we went here on the fly about aroma compounds. Never heard of aroma scalping before. Aroma scalping is when aromas dissolve into especially plastics. It's a big area of science in the food industry. And all the packaging we have in our grocery stores with, say, if you have slices of pineapple in a plastic container, that there's a lot of science behind the container so that the ethyl acetate or whatever it is in the pineapples doesn't get lost and absorbed into the plastic. So we learned about aroma scalping and we said, well, long tubes are not necessarily a good thing. And if they are long tubes, they have to be impermeable to aromas. So we then developed further thinking. This graph in the center and the bottom shows our current machine. It's 1 square foot in diameter. Replace this machine that we wheeled into the winery. So our little machine now has a tri-clamp connection on the bottom. You just hook it up to the outlet of the tank, and gases flow in to the machine and back out to the headspace. It's small, weighs less than 20 pounds, and it can sit on a tank of up to 5,000 liters size. So our little machine that we have now is kind of restricted to fermenting wine volumes up to about 5,000 liters.
And it's one machine per tank.
Yep, that's right. You can take that off when your fermentation is done. Maybe you don't want to use it for the whole fermentation. Remember our discussion about when aromas are produced. Personally, I think it's more effective to use Aromalock from about the peak of CO2 production until the mid to somewhere between the middle to the end of fermentation. If you let it on too long, it doesn't seem to work as well. So you could take that off at some time and put it on another tank right away, plug it in, push the button, and that's it. That's all that's required.
We have 2 more questions. Do you see the same type of aroma stripping during sparging with nitrogen? And will Aromalock work to retain volatile aromas during sparging?
That's a very good question. This graph I showed you of the aroma compounds in different parts of the flow circuit, those aromas are driven out of the experimental tank by CO2. So I sparged CO2 into an artificial solution containing about 13 different aroma compounds, and then we analyze those at the local college lab. But CO2 works very well. There's no reason to think that sparging nitrogen wouldn't work just about as well. In fact, several aroma researchers use nitrogen to sparge aromas from different liquids. Now, could Aromalock be used during sparging with nitrogen? Absolutely. the physics are almost the same, whether it's CO2 or nitrogen carrying the aromas. The aromas will go across our membrane and be pumped back to the headspace. Now, CO2 is more soluble in the membrane and it diffuses more than nitrogen would. So we probably wouldn't get the volume of nitrogen going across the membrane, but we would get some. We need some volume of gas going across the membrane so the vacuum pump has something to pump. If it's just the aroma compounds, we'd have less than a milliliter of gas in the pump circuit, and nothing would flow back to the headspace. So we need some carrier gas, as we call it, to move the aromas back. And CO2 does it in our system, but nitrogen would do it as well.
Okay, thank you. Another question, is the efficiency of the machine affected by temperature?
Well, the efficiency of the machine, yes, it is, but I think if you took your tank with this machine on it and moved it outside in our area where it's snowing today, yes, that would have a hell of an effect on the machine. You may be surprised to learn that sometimes when you cool these membranes down to below room temperature, they actually work better. Sometimes aroma compounds dissolve better in cold things than in warm things. Like, that's the reason cold fermentations work, because the wine is cool, and that tends to retain aromas in it. So the solubility of compounds is affected by temperature, and the solubility of aroma compounds in our membrane is also affected by temperature. Now, we've never tried it except for the normal temperatures of fermentation. So we've used Aromalock at temperatures from, I guess, as low as about 13 degrees C up to 30 degrees C. And we just did a trial in Germany this fall, still waiting for the results to come back. Some of them have trickled back. Geisenheim University in Germany did a study on just that. It looked at the differences in temperature and its effects on the function of the Aromalock system. So the data is there, I just don't have it yet. Germany is really in a lockdown state. And so we did 2 trials in Germany, one at Weisenberg and the other at Geisenheim. Both universities had students working on projects. So we still don't know what the results are. Indications at this point are the wine tastes better, the Romalock wine, and that's it. But they did a study on the effects of temperature on Romalock effectiveness. And I'm just sorry, but we're going to have to wait until we get those data.
Thank you. The next question is, what happens during open air pump overs for red wines? Do you lose some of the benefits of using Aromalock?
Yes, you do. Every time you open the lid, all the headspace gas is probably going to escape. And that includes your concentrated aroma that you pump back. Now, having said that, if you close up the lid tightly again, that increased aroma will regenerate. And the thing is, I can't tell you just how fast it will regenerate. We could probably calculate it. We know what the flow of gas is going back to the headspace. It would depend on the volume of the headspace and so on, but it would reestablish itself. But I would guess that the Romalock would be less effective, but I bet it would still be effective.
Right, thank you. Another question, is the technology available now commercially, and can you give us an idea of price?
Well, yes, we started making these small little machines. We thought that the people who would be interested in this would be the people who try to produce very high quality wine, like the boutique winery, and so they would have relatively small fermentations. So our small machine is for sale, and we have a price tag on that in the $6,000 to $7,000 range at this point.
Thank
We
you.
are in the process of designing a system for up to 30,000 liters, but we don't have it yet.
Thank you. These are all the questions I have so far. So anybody else have any questions? Dietmar, thank you for
I
being
wonder if Jason Santani is on the line, if he could comment.
I am not sure. I tried to reach out to him, but I'm not sure if he is or he isn't, or if he's having trouble with his internet connection.
Yeah, that's probably
Yeah.
a lot of places in Texas. Well, thank you, everybody who participated in this. Thank you for toughing it out and putting up with the environmental things to listen to me. I really appreciate it.
And thank you to everyone around the world. I see, as usual, we have people from all over the world.
Well, that's excellent.
That's very exciting. Please, 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 please take a minute to fill out the survey popping up. Oh, Grimsby. Hi, Michelle. I miss Grimsby. How are you? Oh my goodness. It's so good to see people I used to work with. All right, so please take a minute to fill out the survey at the end of the webinar. With that, I will say thank you all and have a wonderful day. Goodbye for now.